Introduction. All mammals share certain characteristics: milk-producing mammary glands, 3 bones, the middle ear and 1 in the lower jaw, fur or hair, heterodont dentition, different kinds of teeth, and both sebaceous, oil-producing, and sudoriferous, sweat, glands. What about placenta formation during embryonic development? This is a characteristic of humans, as we'll explore in Chapter 3 of MCAT Biology Review, but there are 2 groups of mammals that birth their young a bit differently: prototherians and metatherians. Prototherians, monotremes, which include the duck-billed platypus and echidna, spiny anteater, in case they're developing embryos within hard-shelled amniotic eggs and lay them to be hatched, like reptiles. This method of development is referred to as oviparity. Metatherians, marsupials, include koalas and kangaroos. A typical metatherian fetus, joey, undergoes some development in its mother's uterus and then climbs out of the birth canal and into her marsupium, or pouch. It might seem a bit strange that something as essential as reproduction can be so different between mammalian species, but there are, in fact, a wide variety of reproductive schemes in nature. Many organisms reproduce without a sexual partner. Others can reproduce sexually or asexually depending on environmental conditions. In Chapter 1 of MCAT Biology Review, we explored how bacteria and viruses reproduce. In this chapter, we'll explore eukaryotic and female reproductive systems. 2.1 The cell cycle and mitosis. In animals, autosomal cells are said to be diploid, 2n, which means that they contain 2 copies of each chromosome. Germ cells, on the other hand, are haploid, n, containing only 1 copy of each chromosome. In humans, these numbers are 46 and 23, respectively. We inherit 23 chromosomes from each parent. Eukaryotic cells replicate through the cell cycle, a specific series of phases during which a cell grows, synthesizes DNA, and divides. Derangements of the cell cycle can lead to unchecked cell division and may be responsible for the formation of cancer. Introduction. The cell cycle. The cell cycle is a perennial MCAT favorite. For actively dividing cells, the cell cycle consists of 4 stages: G1, S, G2 and M. The first 3 stages, G1, S, and G2, are known collectively as interphase. Interphase is the longest part of the cell cycle. Even actively dividing cells spend about 90% of their time in interphase. Cells that do not divide spend all of their time in an offshoot of G1 called G0. During the G0 stage, the cell is simply living and carrying out its functions, without any preparation for division. During interphase, individual chromosomes are not visible with light microscopy because they are in a less condensed form known as chromatin. This is because the DNA must be available to RNA polymerase so that genes can be transcribed. During mitosis, however, it is preferable to condense the DNA into tightly coiled chromosomes to avoid losing any genetic material during cell division. G1 stage, pre-synthetic gap. During the G stage, cells create organelles for energy and protein production, mitochondria, ribosomes, and endoplasmic reticulum, while also increasing their size. In addition, passage into the S synthesis stage is governed by a restriction point. Certain criteria, such as containing the proper complement of DNA, must be met for the cell to pass the restriction point and enter the synthesis stage. S stage, synthesis of DNA. During the S stage, the cell replicates its genetic material so that each daughter cell will have identical copies. After replication, each chromosome consists of 2 identical chromatids that are bound together at a specialized region known as the centromere. Note that the ploidy of the cell does not change even though the number of chromatids has doubled. In other words, humans in this stage still only have 46 chromosomes, even though 92 chromatids are present. Cells entering G2 have twice as much DNA as cells in G1. G2 stage, post-synthetic gap. During the G2 stage, the cell passes through another quality control checkpoint. DNA has already been duplicated, and the cell checks to ensure that there are enough organelles and cytoplasm for 2 daughter cells. Furthermore, the cell checks to make sure that DNA replication proceeded correctly to avoid passing on an error to daughter cells that may further pass on the error to their progeny. M stage, mitosis. The M stage consists of mitosis itself along with cytokinesis. Mitosis is divided into 4 phases: prophase, metaphase, anaphase, and telophase. The features of each phase will be discussed in the next section. Cytokinesis is the splitting of the cytoplasm and organelles between the 2 daughter cells. Control of the cell cycle. The cell cycle is controlled by checkpoints, most notably between the G1 and S phase and the G2 and M phase. At the G1/S checkpoint, the cell determines if the condition of the DNA is good enough for synthesis. As mentioned above, this check point is also known as the restriction point. If there has been damage to the DNA, the cell cycle goes into arrest until the DNA has been repaired. The main protein in control of this is p53. At the G2/M checkpoint, the cell is mainly concerned with ensuring that it has achieved adequate size and the organelles have been properly replicated to support 2 daughter cells. p53 also plays a role in the G2/M checkpoint. The molecules responsible for the cell cycle are known as cyclins and cyclin-dependent kinases, CDK. In order to be activated, CDKs require the presence of the right cyclins. During the cell cycle, concentrations of the various cyclins increase and decrease during specific stages. These cyclins bind to CDKs, creating an activated CDK cyclin complex. This complex can then phosphorylate transcription factors. Transcription factors then promote transcription of genes required for the next stage of the cell cycle. Cancer. Cell cycle control is essential to ensure that cells that are damaged or inadequately sized do not divide. When cell cycle control becomes deranged, and damaged cells are allowed to undergo mitosis, cancer may result. One of the most common mutations found in cancer is mutation of the gene that produces p53, called TP53. When this gene is mutated, the cell cycle is not stopped to repair damaged DNA. This allows mutations to accumulate, eventually resulting in a cancerous cell that divides continuously and without regard to the quality or quantity of the new cells produced. Often, cancer cells undergo rapid cell division, creating tumors. Eventually, if the cell begins to produce the right factors, such as proteases that can digest basement membranes or factors that encourage blood vessel formation, the damaged cells are then able to reach other tissues. This may include both local invasion as well as distant spread of cancerous cells through the bloodstream or lymphatic systems. The latter result is known metastasis. Mitosis. Mitosis is the process by which 2 identical daughter cells are created from a single cell. Mitosis consists of 4 distinct phases: prophase, metaphase, anaphase, and telophase, and occurs in somatic cells, or cells that are not involved in sexual reproduction. Prophase. Prophase is the first phase in mitosis. The first step in prophase involves condensation of the chromatin into chromosomes. Also, the centriole pairs separate and move toward opposite poles of the cell. These paired cylindrical organelles are located outside the nucleus in a region known as the centrosome and are responsible for the correct division of DNA. Once the centrioles migrate to opposite poles of the cell, they begin to form spindle fibers, which are made of microtubules. This establishes the centrosome as one of the 2 microtubule organizing centers of the cell, the other being the basal body of a flagellum or cilium. Each of the fibers radiates outward from the centrioles. Some microtubules form asters that anchor the centrioles to the cell membrane. Others extend toward the middle of the cell. The nuclear membrane dissolves during prophase, allowing these spindle fibers to contact the chromosomes. The nucleoli become less distinct and may disappear completely. Kinetochores, which appear at the centrosome, are protein structures located on the centromeres that serve as attachment points for specific fibers of the spindle apparatus, appropriately called kinetochore fibers. Metaphase. In metaphase, the centriole pairs are now at opposite ends of the cell. The kinetochore fibers interact with the fibers of the spindle apparatus to align the chromosomes at the metaphase plate, equatorial plate, which is equidistant from the two poles of the cell. Anaphase. During anaphase, the centromeres split so that each chromatid has its own distinct centromere, thus allowing the sister chromatids to separate. The sister chromatids are pulled toward the opposite poles of the cell by the shortening of the kinetochore fibers. Telophase and cytokinesis. Telophase is essentially the reverse of prophase. The spindle apparatus disappears. A nuclear membrane reforms around each set of chromosomes, and the nucleoli reappear. The chromosomes uncoil, resuming their interphase form. Each of the two new nuclei receives a complete copy of the genome identical to the original genome and to each other. Cytokinesis, which occurs at the end of telophase, is the separation of the cytoplasm and organelles, giving each daughter cell enough material to survive on its own. Each cell undergoes a finite number of divisions before programmed death. For human somatic cells, this is usually between 20 and 50. After that, the cell can no longer divide continuously. Meiosis. Whereas mitosis occurs in somatic tissue and results in two identical daughter cells, meiosis occurs in gametocytes, germ cells, and results in up to 4 non-identical sex cells, gametes. Meiosis shares some similarities with mitosis. In both processes, for instance, genetic material must be duplicated, chromatin is condensed to form chromosomes, and microtubules emanating from centrioles are involved dividing genetic material. However, the MCAT tends to ask about the differences between these two processes. In contrast to mitosis, which consists of one round each of replication and division, meiosis consists of one round of replication followed by 2 rounds of division. Meiosis I result in homologous chromosomes being separated, generating haploid daughter cells. This is known as reductional division. Meiosis II is similar to mitosis, in that it results in the separation of sister chromatids without change in ploidy, and is therefore known as equational division. Meiosis I. The human genome is composed of 23 homologous pairs of chromosomes, homologs, each of which contains one chromosome inherited from each parent. This brings up an important note about terminology. Whereas homologous pairs are considered separate chromosomes, such as maternal chromosome 15 and paternal chromosome 15, sister chromatids are identical strands of DNA connected at the centromere. Thus, after S phase, there are 92 chromatids organized into 46 chromosomes, which are organized into 23 homologous pairs. Prophase 1. During prophase 1, the chromatin condenses into chromosomes, the spindle apparatus forms, and the nucleoli and nuclear membrane disappear. The first major difference between meiosis and mitosis occurs at this point. Homologous chromosomes come together and intertwine in a process called synapsis. At this point, each chromosome consists of 2 sister chromatids, so each synaptic pair contains 4 chromatids and is referred to as a tetrad. The homologous chromosomes are held together by a group of proteins called the synaptonemal complex. Chromatids of homologous chromosomes may break at the point of contact, called the chiasma, plural, chiasmata, and exchange equivalent pieces of DNA. This process is called crossing over, and can be characterized by the number of crossover events that occur in one strand of DNA, including single crossovers and double crossovers. Note that crossing over occurs between homologous chromosomes and not between sister chromatids of the same chromosome. The latter are identical, so crossing over would not produce any change. Those chromatids involved are left with an altered but structurally complete set of genes. Such genetic recombination can unlink linked genes, thereby increasing variety of genetic combinations that can be produced via gametogenesis. Linkage refers to the tendency for genes to be inherited together. Genes that are located farther from each other physically are less likely to be inherited together, and more likely to undergo crossing over relative to each other. Thus, as opposed to asexual reproduction, which produces identical offspring, sexual reproduction provides the advantage of great genetic diversity, which is believed to increase the ability of a species to evolve and adapt to a changing environment. Because of crossing over, each daughter cell will have a unique pool of alleles, genes coding for alternative forms of a given trait, from random mixture of maternal and paternal origin. In classical genetics, crossing over explains Mendel's second law, of independent assortment, which states that the inheritance of one allele has no effect on the likelihood of inheriting certain alleles for other genes. Metaphase 1. During metaphase 1, homologous pairs, tetrads, align at the metaphase plate, and each pair attaches to a separate spindle fiber by its kinetochore. Note the difference from mitosis. In mitosis, each chromosome is lined up on the metaphase plate by two spindle fibers, one from each pole. In meiosis, homologous chromosomes are lined up across from each other at the metaphase plate and are held by one spindle fiber. Anaphase 1. During anaphase 1, homologous pairs separate and are pulled to opposite poles of the cell. This process is called disjunction, and it accounts for Mendel's first law, of segregation. During disjunction, each chromosome of paternal origin separates, or disjoins, from its homologue of maternal origin, and either chromosome can end up in either daughter cell. Thus, the distribution of homologous chromosomes to the two intermediate daughter cells is random with respect to parental origin. This separating of the two homologous chromosomes is referred to as segregation. Telophase 1. During telophase 1, a nuclear membrane forms around each new nucleus. At this point, each chromosome still consists of two sister chromatids joined at the centromere. The cells are now haploid. Once homologous chromosomes separate, only N chromosomes are found in each daughter cell, 23 in humans. The cell divides into two daughter cells by cytokinesis. Between cell divisions, there may be a short rest period, or interkinesis, during which the chromosomes partially uncoil. Meiosis 2. Meiosis 2 is very similar to mitosis in that sister chromatids rather than homologues are separated from each other. Prophase 2. During prophase 2, the nuclear envelope dissolves, nucleoli disappear, the centrioles migrate to opposite poles, and the spindle apparatus begins to form. Metaphase 2. During metaphase 2, the chromosomes line up on the metaphase plate. Anaphase 2. During anaphase 2, the centromeres divide, separating the chromosomes into sister chromatids. These chromatids are pulled to opposite poles by spindle fibers. Telophase 2. During telophase 2, a nuclear membrane forms around each new nucleus. Cytokinesis follows, and two daughter cells are formed. Thus, by completion of meiosis 2, up to 4 haploid daughter cells are produced per gametocyte. We use the phrase up to because oogenesis, discussed later in this chapter, may result in fewer than 4 cells if an egg remains unfertilized after ovulation. The reproductive system. Biological sex is determined by the 23rd pair of chromosomes, with XX being female and XY being male. Ova can only carry the X chromosome, while sperm can of genetic information. Mutations in these genes can cause sex-linked, X-linked, disorders. Males are termed hemizygous with respect to many of the genes on the X chromosome because they only have one copy. Therefore, a male with a disease carry either the X or Y chromosome. The X chromosome carries a sizable amount, causing allele on the unpaired part of X chromosome will necessarily express that allele. Females, on the other hand, may be homozygous or heterozygous with respect to genes on the X chromosome. Most X-linked disorders are recessively inherited, therefore, females express these disorders far less frequently than males. Females carrying a diseased allele on an X chromosome but not exhibiting the disease are said to be carriers. Comparatively, the Y chromosome contains very little genetic information. One notable gene on the Y chromosome is SRY, sex-determining region Y, which codes for a transcription factor that initiates testis differentiation and, thus, the formation of male gonads. Therefore, in the absence of the Y chromosome, all zygotes will be female. In the presence of the Y chromosome, a zygote will be male. Male reproductive anatomy. In males, the primitive gonads develop into the testes. The testes have two functional components, the seminiferous tubules and the interstitial cells of Leydig. Sperm are produced in the highly coiled seminiferous tubules, where they are nourished by Sertoli cells. The cells of Leydig secrete testosterone and other male sex hormones, androgens. The testes are located in the scrotum, an external pouch that hangs below the penis, a position that allows it to maintain a temperature 2 degrees to 4 degrees Celsius lower than the body. In fact, there is a layer of muscle around the vas deferens, ductus deferens, that can raise and lower the testis to maintain the proper temperature re-sperm development. As sperm are formed, they are passed to the epididymis, where their flagella gain motility, and they are then stored until ejaculation. During ejaculation, sperm travel through the vas deferens and enter the ejaculatory duct at the posterior edge of the prostate gland. The two ejaculatory ducts then fuse to form the urethra, which carries sperm through the penis as they exit the body. In males, the reproductive and urinary systems share a common pathway. This is not the case in females. As sperm pass through the reproductive tract, they are mixed with seminal fluid, which is produced through a combined effort by the seminal vesicles, prostate gland, and bulbourethral gland. The seminal vesicles contribute fructose to nourish sperm. And both the seminal vesicles and prostate gland give the fluid mildly alkaline properties so the sperm can survive in the relative acidity of the female reproductive tract. The bulbourethral, Cooper's, glands produce a clear viscous fluid that cleans out any remnants of urine and lubricates the urethra during sexual arousal. The combination of sperm and seminal fluid is known as semen. Spermatogenesis. As mentioned above, spermatogenesis, the formation of haploid sperm through meiosis, occurs in the seminiferous tubules. In males, the diploid stem cells are known as spermatogonia. After replicating their genetic material, S stage, they develop into diploid primary spermatocytes. The first meiotic division will result in haploid secondary spermatocytes, which then undergo meiosis II to generate haploid spermatids. Finally, the spermatids undergo maturation to become mature spermatozoa. Spermatogenesis results in 4 functional sperm for each spermatogonium. Mature sperm are very compact. They consist of a head, containing the genetic material, a midpiece, which generates ADP from fructose, and a flagellum, for motility. The midpiece is filled with mitochondria, which generate the energy for swimming through the female reproductive tract to reach the ovum in the fallopian tubes. Each sperm head is covered by cap known as an acrosome. This structure is derived from the Golgi apparatus and is necessary to penetrate the ovum. Once a male reaches sexual maturity during puberty, approximately 3 million sperm are produced per day for the rest of his life. Female reproductive anatomy. Unlike the male reproductive system, all of the female reproductive organs are internal. The gonads, known as ovaries, produce estrogen and progesterone. The ovaries are located in the pelvic cavity. Each consists of thousands of follicles, which are multilayered sacs that contain, nourish, and protect immature ova, eggs. Between puberty and menopause, 1 egg per month is ovulated into the peritoneal sac, which lines the abdominal cavity. It is then drawn into the fallopian tube or oviduct, which is lined with cilia to propel the egg forward. The fallopian tubes are connected to the muscular uterus, which is the site of fetal development. The lower end of the uterus, known as the cervix, connects to the vaginal canal, where sperm are deposited during intercourse. The vagina is also the passageway through which childbirth occurs. The external female anatomy is known collectively as the vulva. As mentioned earlier, females have separate excretory and reproductive tracts. Oogenesis. The production of female gametes is known as oogenesis. Although gametocytes undergo the same meiotic process in both females and males, there are some significant differences between the two sexes. First, there is no unending supply of stem cells analogous to spermatogonia in females. All of the oogonia a woman will ever have are formed during fetal development. By birth, all of the oogonia have already undergone DNA replication and are considered primary oocytes. These cells are 2N, like primary spermatocytes, and are actually arrested in prophase I. Once a woman reaches menarche, her first menstrual cycle, 1 primary oocyte per month will complete meiosis I, producing a secondary oocyte and a polar body. The division is characterized by unequal cytokinesis, which distributes ample cytoplasm to one daughter cell, the secondary oocyte, and nearly none to the other, the polar body. The polar body generally does not divide any further and will never produce functional gametes. The secondary oocyte, on the other hand, remains arrested in metaphase II and does not complete the remainder of meiosis unless fertilization occurs. Oocytes are surrounded by 2 layers, the zona pellucida and the corona radiata. The zona pellucida surrounds the oocyte itself and is an acellular mixture of glycoproteins that protect the oocyte and contain compounds necessary for sperm cell binding. The corona radiata lies outside the zona pellucida and is a layer of cells that adhered to the 0-OCYT during ovulation. Meiosis II is triggered when a sperm cell penetrates these layers with the help of acrosomal enzymes. The secondary oocyte undergoes the second meiotic division to split into a mature ovum and another polar body, which will eventually be broken down. A mature ovum is a very large cell consisting of large quantities of cytoplasm and organelles. The ovum contributes nearly everything to the zygote, half of the DNA and all of the cytoplasm, organelles including mitochondria, and RNA for early cellular processes, whereas sperm only contribute half of the DNA. Upon completion of meiosis II, the haploid pronuclei of the sperm and the ovum join, creating a diploid zygote. Sexual development. The ability to reproduce is under hormonal control. Prior to puberty, the hypothalamus restricts production of gonadotropin-releasing hormone, GnRH. At the start of puberty, this restriction is lifted as the hypothalamus releases pulses of GnRH, which then triggers the anterior pituitary gland to synthesize and release follicle-stimulating hormone, FSH, and luteinizing hormone, LH. These hormones trigger the production of other sex hormones that develop and maintain the reproductive system. Male sexual development during the fetal period, from 9 weeks after fertilization until birth, presence of the Y chromosome leads to production of androgens, resulting in male sexual differentiation. For the duration of infancy and childhood, androgen production is low. Testosterone, produced by the testes, increases dramatically during puberty, and sperm production begins. In order to achieve this, there is a delicate interplay of FSH and LH stimulation on 2 cell types in the testes. FSH stimulates the Sertoli cells and triggers sperm maturation, whereas LH causes the interstitial cells to produce testosterone. Testosterone not only develops and maintains the male reproductive system, but also results in the development of secondary sexual characteristics such as facial and axillary hair, deepening of the voice, and increased muscle and bone mass. Testosterone production remains high into adulthood and declines as men age. This hormone exerts negative feedback on the hypothalamus and anterior pituitary so that production is kept within an appropriate range. Female sexual development. The ovaries, which are derived from the same embryonic structures as the testes, are also under the control of FSH and LH secreted by the anterior pituitary. The ovaries produce estrogens and progesterone. Estrogen are secreted in response to FSH and result in the development and maintenance of the female reproductive system and female secondary sexual characteristics, breast growth, widening of the hips, changes in fat distribution. In the embryo, estrogens stimulate development of the reproductive tract. In adults, estrogens lead to the thickening of the lining of the uterus, endometrium, each month in preparation for the implantation of a zygote. Progesterone is secreted by the corpus luteum, the remains of the ovarian follicle following ovulation in response to LH. Interestingly, progesterone is involved in the development and maintenance of the endometrium, but not in the initial thickening of the endometrium. This is the role of estrogen. This means that both estrogen and progesterone are required for the generation, development, and maintenance of an endometrium capable of supporting a zygote. By the end of the first trimester of a pregnancy, progesterone is supplied by the placenta, while the corpus luteum atrophies and ceases to function. The menstrual cycle. During the reproductive years, from menarche to menopause, estrogen and progesterone levels rise and fall in a cyclic pattern. In response, the endometrial lining will grow and be shed. This is known as the menstrual cycle and can be divided into 4 events: the follicular phase, ovulation, the luteal phase, and menstruation. Follicular phase. The follicular phase begins when the menstrual flow, which sheds the uterine lining of the previous cycle, begins. GnRH secretion from the hypothalamus increases in response to the decreased concentrations of estrogen and progesterone, which fall off toward the end of each cycle. The higher concentrations of GnRH cause increased secretions of both FSH and LH. These two hormones work in concert to develop several ovarian follicles. The follicles begin to produce estrogen, which has negative feedback effects and causes the GnRH, LH, and FSH concentrations to level off. Estrogen stimulates regrowth of the endometrial lining, stimulating vascularization and glandularization of the decidua. Ovulation. Estrogen is interesting in that it can have both negative and positive feedback effects. Late in the follicular phase, the developing follicles secrete higher and higher concentrations of estrogen. Eventually, estrogen concentrations reach a threshold that paradoxically results in positive feedback, and GnRH, LH, and FSH levels spike. The surge in LH is important. It induces ovulation, the release of the ovum from the ovary into the abdominal, peritoneal, cavity. Luteal phase. After ovulation, LH causes the ruptured follicle to form the corpus luteum, which secretes progesterone. Remember that estrogen helps regenerate the uterine lining, but progesterone maintains it for implantation. Progesterone levels begin to rise, while estrogen levels remain high. The high levels of progesterone cause negative feedback on GnRH, FSH, and LH, preventing the ovulation of multiple eggs. Menstruation. Assuming that implantation does not occur, the corpus luteum loses its stimulation from LH, progesterone levels decline, and the uterine lining is sloughed off. The loss of high levels of estrogen and progesterone removes the block on GnRH so that the next cycle can begin. Pregnancy. On the other hand, if fertilization has occurred, the resulting zygote will develop into a blastocyst that will implant in the uterine lining and secrete human chorionic gonadotropin, HCG, an analog of LH. It looks very similar chemically and can stimulate LH receptors. This maintains the corpus luteum. HCG is critical during first trimester development because the estrogen and progesterone secreted by the corpus luteum keep the uterine lining in place. By the second trimester, HCG levels decline because the placenta has grown to a sufficient size to secrete enough progesterone and estrogen by itself. The high levels of estrogen-progesterone continue to serve as negative feedback on GnRH secretion. Menopause. As woman ages, her ovaries become less sensitive to FSH and LH, resulting in ovarian atrophy. As estrogen and progesterone levels drop, the endometrium also atrophies, and menstruation stops. Also, because the negative feedback on FSH and LH is removed, the blood levels of these two hormones rise. This is called menopause. Profound physical and physiological changes usually accompany this process, including flushing, hot flashes, bloating, headaches, and irritability. Menopause usually occurs between the ages of 45 and 55. Conclusion. In this chapter, we explored one of the key tenets of the cell theory: how cells produce more copies of themselves. We first examined mitosis, which results in genetically identical diploid daughter cells. We then moved on to meiosis, which results in genetically non-identical haploid daughter cells, or gametes. We then looked at the male and female reproductive systems, which form these gametes, each of which contains half of the normal complement of genetic information. Finally, we explored basic reproductive endocrinology and saw how testosterone and estrogen are key in the development of the reproductive systems and the secondary sex characteristics that develop at puberty. Formation of gametes is only half the story, of course. It serves us no good as a species to form sex cells if the cells cannot interact to form another human. Ultimately, gametes must accomplish their purpose, passing on the genes, the instructions for life, from one generation to another. Thus, we turn our attention in the next chapter to the next steps of fertilization, embryogenesis, and birth. The development of another human is a humbling and inspiring process. Indeed, it is through the union of one egg and one sperm that every human being on the planet today and since the beginning of the human race came into existence.